Hi, everyone. So my name is Nick D'Arienzo, and today I'm going to be telling you about work that I did with Dr. Anna Dornhaus, as well as Dr. Pierre-Olivier Montiglio, during my time here as a postdoctoral fellow at the University of Arizona. This work is looking at stuff that I've developed over the last few years, looking at how individual variation across multiple traits, specifically behavior and extended phenotypic traits, web structure in this case, interact to actually determine success. And so I think like two fundamental questions that probably most people are interested in are like A, why are traits correlated? And B, why is there diversity within traits? And the phenotypic integration literature can kind of actually address both of this. So the idea behind phenotypic integration is that animals should evolve correlated suites of behavior that span from genetics to physiology to their underlying neural architecture to behavior and morphology, as these traits need to interact with one another in order for an animal to perform. And so this very much makes sense. If you're going to be aggressive, you need the associated morphology and musculature to be aggressive and back up that behavior. And so this is typically applied like across species, but it actually works well within species to explain why we see suites of correlated traits. And so I think a great example is Brody's 1992 study in MNAT, looking at within species variation of garter snakes, so how these polymorphisms and coloration could be maintained. So you see both striped morphs exist in the population, but then you also see non-striped sort of spotted morphs to even then just ones with no coloration. And so what Brody found is that the different color morphs can have similar fitness outcomes over the course of a year, not being eaten, depending on what behavior they perform when the predator's around. So if you're spotted and performed reversals when you had a predator cue, you had similar fitness as if you were striped and didn't perform reversals. The underlying idea being that you need behavior to match your coloration in order to reduce how conspicuous you are. One doesn't function without the other. And so this can show both why traits are correlated as well as why there's diversity within these traits. But again, a lot of this literature is actually focused more on a lot of morphological traits and some physiological, and not always integrating as much behavior and let alone these extended phenotypic traits, which I think are probably a big thing underlying a lot of this and are an interesting thing to look at. So I think that these extended phenotypic traits are the likely dimensions of this integrated phenotype for a number of reasons. Just kind of broadly speaking, they're found across multiple taxa. So here we have, a bowerbird's nest, and then of course social insects, make these very elaborate nests that are used for a lot of things. And so again, in terms of what they're used for, they perform a ton of different functions, from foraging and anti-predator behavior to also things like sexual selection and thermal regulation. Sometimes they perform only a single one. In the case of bowerbirds, the males are just courting from this. But in social insects, these are doing everything. And so these are doing things that also overlap with behavior and behavior much work with. So it makes sense these are probably actually integrated into this phenotypic whole, but we don't quite know how and if they are interacting differently or independently from these behavioral traits. Furthermore, there's known individual variation. So we know now there's a lot of individual variation in behavior in this, but there's also known individual variation in a lot of these phenotypic traits. So male three-spine sticklebacks vary in terms of how much they build these grass nests. And then temnothorax rock ants, so here colonies you can see build dramatically different nests over both time and different contexts. Some having these thicker lines right here, but on the lower picture you see that they're building more sparse dispersed walls. So individual variations known to exist, and presumably something maintaining this variation that might be interacting with the phenotypic whole. And there's actually evidence to suggest that these do very much interact with behavioral expression. So a wonderful study by Noah Pinter-Woolman, what it demonstrated is that in harvester ants, as colonies moved across and migrated from different nest sites from one to another, they didn't maintain consistency in their colony level behavior. But as colonies then over time repeatedly occupied the same nest site, so different colonies in the same nest, that nest site elicited consistent differences in behaviors in those colonies, implying that the structure of the nest itself is what's dictating colony performance, or at least behavioral expression, not the colony itself. Structure of the nest is. Similarly, Pierre-Olivier and I did a study looking at how the web structure of a black widow spider determines behavioral response. And so we swapped widow spiders around in different webs, and what we found is that the idea of the web alone explained 25% of the behavioral variation. Again, highlighting the fact that these are likely this dimension of the phenotypic whole that are highly integrated, thus me getting excited about studying these things. So there's two questions that I'm going to kind of generally focus on here and then talk about a more specificity in the context of the system. One, I'm really interested in sort of why behavioral how and if they do interact and influence fitness? And two, can these different types of interactions maintain variation with the population? So now I want to just talk to you about black widow spiders as a system. So black widows are great. One of the reasons is there's known variation both behavioral and phenotype. Some are consistently more likely to attack a prey cue than other ones. And then they also differ in their web structure. So some, if you look here on the bottom left picture, consistently build more of the gum-footed lines GF, which have then this sticky silk at the bottom end that helps get in prey capture, where others then consistently build more structural lines. And so those are the ST lines that then connect to the bottom of the ground ultimately. And these are used for safety, that then provide more protection from potentially predators and other spiders. There's individual differences there. They're also really easy to assess. You can put spiders in a box and let them then build their webs, and you can count the number of lines that connect to the ground. So you can rapidly assess this over multiple trials. It's also really easy to assess their behavior. So you can just apply a vibratory cue to the web, and the spiders will either run out and attack it if it's an actual prey item or not. So we can do this repeatedly multiple times without altering their state or experience. It's just they're either getting the prey cue or they're not, and then measuring their response. And the other thing that I think makes these really great in terms of just in general trying to study these correlated traits is you can manipulate web structure. So if we're assuming that these traits are correlated one way or another, such as we might assume maybe multiple behavioral traits correlated, the nice part about looking at web structure is we can transplant spiders onto different webs with known structures, or we can modify aspects of the structure they're building on, or even modify the web itself by cutting out specific line types without any adverse effects to the spider. And this is great because if you're trying to do sort of some behavior morphology, if you try to manipulate those, you're putting on a lot of downstream effects of because you're going to have to some way or another affect otherwise. You're looking at modify, say, aggressive behavior. So I think these are a really great tool in general to study a lot of these adaptive nature of correlated traits. So again, in the context of the system, the two questions I'm going to look at for this study is one, how foraging behavior and web structure interact to influence fitness. So, here we have a spider that's responding to an ant that's caught down in the web, you can see. And so how that gum-footed line that is capturing that ant is working with this spider's behavioral response to capture that prey. Does one need the other, or are they able to function independently at one another? And then also, can we see that sort of these non-aggressive spiders that don't build many gum-footed lines, can they have similar prey capture success through some maybe different mechanisms that we're not measuring as aggressive ones? So can these different types actually have similar success, potentially making variation? So the experimental design for this study, what we did, so this is a top-down view of this cardboard structure that we let the spider build upon. You can see on that left rim, there's a retreat for it to go under. So we allow them to build a web for 7 days in this box. After which then we go and count the number of gum-footed lines, so in structural lines, we know the general web makeup. And then we assay their behavior 3 times within a day, and then across 3 days. And then we can sum this up to get a total aggression score. If they attacked all 9 times, they're really aggressive, and they have an attack number of 9. Of course, if they attack just once, well, then they're not aggressive, they have an attack number of 1. So we do this, and then what we do is we remove all the spiders from their webs. We used about 35 spiders in total. All of them got removed from their webs. And then half were put back in their own web and half went to a different web. And so this way we have the web structure, we know what structure they built, and we know what structure they're on and how these differ. We then added in 5 harvester ants, and we counted and then replenished at 1 hour, or counted how many were eaten at 1 hour, 24, 48, 72, and 96 hours. And then we replenished back to 5 each interval. So we had different or equal sampling intervals and equal probability of capturing prey at each interval. After that 96-hour time point, we removed them all, re-assayed the web to see if it changed at all. We weighed the spiders, moved them home, and then fed them to crickets. So then this way we equalized state and let them rest and feed for 7 days. After which then they built a web again, and this whole process started again. The key thing here is that if they started this first round by foraging on their own web, the next round they subsequently went and foraged on a different web. To where if they forage on a different web this first round, then this next round they forage on their own web. And so we did a total of 4 rounds, allowing all spiders to forage twice on their own webs and twice on different webs. Kind of keeping in mind, and I'll just mention quickly, but I don't present on this, we didn't see any effect of the treatment on what they captured. So transplanted spiders were capturing an equal number of prey at a population level as non-transplanted ones. And they also didn't modify their webs one or another. So it's not like any of the results are driven by a transplanted spider quickly modifying the web structure to match their own. They kept the same web structure. So what do we see in results overall? So in terms of trait correlations, we see things that we've seen before, but I'm just going to present them again so you know. So for example, the number of gum-footed lines on our y-axis and the number of structural lines on our x-axis, we see a negative relationship here. Individuals are kind of either building lots of gum-footed lines with structural or vice versa. And this relates to a behavior in a way that we, again, have seen before. If you build a lot of gum-footed lines, you tend to be much more aggressive. If you build few, you tend to be less aggressive. And then structural lines, if you build more of those, you also tend to be less aggressive. And so you can kind of view this that there's two different types. So we have aggressive individuals with many gum-footed lines, and we have non-aggressive individuals that build many structural lines. So an aggressive foraging strategy versus a more safer strategy, and of course, continuous variation between. So looking at now how these things actually interact to influence prey capture, what we did was we did model comparison, which allows us to compare models with different structures. And we find the best fit model actually didn't include an interaction between behavior and web structure. What we found was that these independently contributed to prey capture. So on our y-axis here, we have the proportion of ants within a time interval. And on our x-axis, we have the number of gum-footed lines present on that web. And so it was just positive related. If you have more gum-footed lines, you're more effective at capturing prey. Aggression is the same way. So again, we have that same y-axis, but now attack number. If you attack that prey queue more, you're also capturing more prey when actually presented with it in a live foraging context. So again, the highlight here is that these appear to be both contributing independently and maybe not be interacting. We're also controlling for being on a different web. But the key thing is, the whole point of this experiment is that we transplanted them to actually look directly if these are interacting rather than having to infer it through this. And so now we can look within just the swap spiders. And so swapping them reveals that these traits very much interact with one another. So here again, we have the same y-axis, or proportion of ants in the interval, but on our x-axis, we have the difference in gum-footed lines in the own web. So if you built a lot of gum-footed lines on your own and then got transplanted to a web with very few, you're always going to have a negative value there. And so this black line here represents just the average level of aggression in the population. So an attack number of around like 4 or 5. And what we see is that same kind of positive relationship as before, is that more gum-footed lines help. But when we plot the interaction with the aggression and gum-footed differences, what we see is that non-aggressive individuals are actually suffering from more gum-footed lines. If you're not aggressive and you go into a web with more, you capture fewer prey. And if you go into a web with fewer gum-footed lines, you capture more prey. Where aggressive ones are very much benefiting from the same or more gum-footed lines. So what this is saying is that these aggressive individuals very much need these specific web structural elements in order to capture prey. You can't actually use your behavior if you don't have the web structure to back it up. And so they might not build an optimal number for their behavioral phenotype, but they could be constrained by state, or there could be a lot of interactions going on. What's interesting is that we also see a similar thing happening in structural lines. Aggressive individuals suffer from having more structural lines and benefit when there's fewer, so they don't want these getting in the way properly. The exact mechanism is unsure. And then non-aggressive ones benefit, generally speaking, or a little bit from having more structural lines. And so this highlights that, the behavior very much benefits from this sort of match web structure, and that you can have this similar success also across the spectrum. We see similar foraging success across these different combinations of traits. So the takeaway points from this are, one, extended phenotypic traits very much correlate with behavioral traits. As we've seen before, and again, emphasizing this, that this is likely a common thing. Individuals also have the highest success when their extended phenotype, web structure, matches their behavioral phenotype. So again, this is emphasizing that these are likely highly integrated and correlated traits. And so correlational selection over time could then drive these trait correlations that we see exist. Similarly, extreme ends can have similar and high success. And so this could ultimately, again, drive the evolution of these genetic correlations, but also maintain variation within this population if the aggressive and non-aggressive individuals are having similar prey capture success. And finally, I think that this emphasizes this notion that, the value of studying these extended phenotypic traits as part of a whole phenotype. Remember sort of these initial results, or the first ones I presented, just showing the positive effect of non-interacting effect of gum-footed lines and attack number. If that's all we had, we would have actually left this study thinking that these are independently contributing success and are not correlated, or the correlation is not what matters. We also would have thought that there would have just been directional selection on aggression and gum-footed lines. And in reality, that's not the case. We see that this phenotypic variation being maintained in different combinations can be again, without studying these things together and actually manipulating these, we would have had a false understanding of the consequences of this trait variation. So thank you for your time. I want to sort of acknowledge a whole host of funders along the way. Of course, people that collaborated on this. I also want to point out two undergraduates that were very critical in this experiment, Charles Bradley and Connor Smith. They both helped a tremendous amount, both in the